Welcome, everyone. I'm Bernadette Toner, Executive Editor at GenomeWeb, and I'll be your moderator for today's Genome webinar. The title of today's session is Predicting Drug Toxicity and Efficacy with Human Primary Blood Cell Models. Our sponsor is Discovery Life Sciences. Our speaker today is Gary Dos Santos, Director of Cell Biology Services at Discovery Life Sciences.
Thanks for the introduction. Just to add a little bit more, I run the Discovery Life Sciences Seattle lab, which is formerly ReachBio, which is specialized in developing and offering on a contract basis toxicity and efficacy models focused almost exclusively on human primary cells, so bone marrow, PBMCs, fresh whole blood, that sort of thing. And we'll be talking today about some of the models that we've developed and our emphasis on clinical translatability. Okay, so a little bit of background, as I'm sure many people on this call already know, but we'll go through it just to frame up the conversation. A lot of programs fail. They go all the way to the clinic and fail because what they work out in their preclinical workup and IND enabling work just doesn't translate fully to the clinic. We're always going to have that challenge, I think, in drug development, but the objective is to create over time more and more biologically relevant and clinically translatable models that can be offered up earlier and earlier in the drug development process to really weed out those candidates that are not going to be successful in the clinic. There's a definite need. We've been doing this for over 20 years, but there is still a need to continue to develop human-based models that are clinically relevant, that reduce the downstream attrition. Try to get that attrition early so that you're not spending those millions and millions of dollars developing drugs that maybe never even have a chance once they get into the clinic. And we've worked with over 800 companies over the past 20 years, seen many, many programs. And one trend that I've noticed in all of them is the ones that are really successful is that the programs, it doesn't even necessarily, it's not the company itself, but the program leadership, they get to the hard questions early. They're really trying to drive attrition of their drugs so they can weed out the poor candidates and get to the really strong candidates early and then move those in. And our goal is, from the hematology perspective, is to help them ask those sometimes challenging questions and identify the best candidates to move forward. All right, and then just in way of a little more background, there is obviously a very strong shift by regulatory agencies, including the FDA, towards new approach methodologies. So these are approaches that replace or enhance animal models in a way that improves clinical predictability and speeds up drug development. The field is moving very, very fast. The timeline here, typical FDA timelines might go back 30 or 40 years. The timeline here really kicks off in April of 2025. And they've been putting out updates and guidance documents continuously really since then. So the FDA is very much invested in moving towards primary cell models, but we need to be careful when we're doing this that we don't simply dismiss animal models. I think there's always going to be a role for animal models in drug development. And then the other thing I want to make sure that we cover is that a new approach methodology is not a single assay. To take something to recapitulate an entire biological process in a Petri dish or a 96-well plate is almost impossible. You can recapitulate components of it, but really what we focus on is trying to get all the way from a hematopoietic stem cell to a mature activated cell using a series of overlapping assays, one that focuses on progenitors, which we'll talk to, one that focuses on more development, and then models that focus on the activation, the end state activation, and investigating what the effect of drugs are there. Okay, we all know, I mean, if you've been in the industry long enough, you know that animal models are important, but there are a lot of challenges associated with them. The high cost, obviously. There's you always have to go through an IACUC to get every experiment or series of experiments approved. It can take quite a long time to execute and then analyze animal data. And one thing I've noticed since we started 20 years ago, since I started the industry 20 plus years ago, is that the modalities and targets that are being developed as time goes on, they're becoming more and more human specific. So some of the relevancy of the animal models has dropped. So for example, 5-fluorouracil, an old drug that's still being used in some cases today. It's going to impact a mouse, it's going to impact a rat, it's going to impact a human, an NHP, and a dog, all in largely similar ways. But newer drugs might be focusing on receptors that are unique to human cells or subsets of human cells. And so it's hard to pick up efficacy or toxicity using the animal models. And so I think that's driving a lot of the need for these human primary cell models, both normal cells to evaluate toxicity and on disease cells to evaluate efficacy. Okay, so like I said before, the earlier we can get to those hard questions accurately and in a clinically translatable way, the better off we are as a program and as an industry. We become more efficient and we avoid, we reduce the number of high profile failures in the clinic. So from our perspective, when we're looking at hematotoxicity, we're looking to answer questions like, will the drug damage bone marrow? And when we say, will it damage bone marrow is a highly complex organ in and of itself. There are blood stem cells, the classic CD34 cell. There's stromal cell, the mesenchymal stem cells. There's the marrow itself. What we're focusing on is really the impact of the drugs on the cells, largely the CD34 derived cells and occasionally the mesenchymal cells. Our models look at toxicity, but we also can modify some of our models to evaluate if toxicity is reversible, for example, by doing washout experiments. And because we develop both toxicity and efficacy models, we can often help in the establishment of an in vitro therapeutic index, looking at the difference in the IC50 on a disease cell versus the IC50 on a healthy cell, an analogous healthy cell. And the objective, of course, is to develop as large an difference in IC50 between the healthy and the diseased cells. If we don't do a good job of predicting hematotoxicity early on, we can get all the way to the clinic, encounter unexpected dose-limiting toxicity, specifically usually around neutropenia and thrombocytopenia, which can lead to study delays and ultimately, in a worst-case scenario, program termination. And that's a very expensive and high-profile failure, obviously, when you get all the way into clinical trials and the drug is not working. Like I said before, sometimes it's unavoidable. You simply can't recapitulate all the complexity of human biology in the preclinical space, but we want to do as good a job as possible before we get into those trials. Okay. So on the topic of differences between human models and animal models, some time ago we sat down and we started examining the difference in some of our in vitro models between species. So we ran an assay called the CFU-GM assay. That is an assay that is specifically designed to look at myeloid progenitor toxicity, which is a major cause of hematotoxicity clinically. We looked at 5 different animal species: human, NHP, dog, rat, and mouse. And we looked at the IC50s in the CFU-GM model for relatively non-toxic or drugs that don't have a large hematotoxic liability clinically, like erlotinib and sorafenib. And we looked the spectrum all the way down to drugs that were fairly toxic, like dasatinib. What we found was that in vitro, when we ran these assays, the human and the NHP cells tended to cluster together on the IC50s. The human, the NHP, and the canines. And then the rat and the mouse would tend to cluster together. So there's the rat and the mouse are in the blue and the green, and then the NHP, human, and canine are down here. Okay. So this is good news in some ways, and it's bad news in others. One is that we can relatively easily at this point take human bone marrow, run it in the CFU-GM assays, and measure an IC50. The bad news is, how do we marry that data with what we're going to see in the mouse models? Because like I say, I don't think we're going to fully get rid of the mouse models ever. And we're going to need to understand how to make that data work together. Okay. And then just before we get into a little more of the details on what I was talking before between how to use animal models with human models, and talk just to drive home, what are we trying to do? We're trying to evaluate with our in vitro models the risk of specific lineage blood toxicity, asking questions. Does the drug interfere with neutrophil development? If it does, you have a potential clinical liability with neutropenia, which will appear very, very quickly upon dosing of patients. And if you hit grade 3 or grade 4, that could stop your program, certainly going to stop the dosing of the patient. And if you're taking out a progenitor, it can take quite some time for the neutrophils to come back. They'll drop off very quickly. But if you've taken out the progenitors, then the stem cells are going to have to regenerate the progenitors, which are going to have to regenerate the neutrophils before everything gets back up and running again. And there can be serious clinical challenges there. Similarly, you can take out the erythrocyte progenitors, which will manifest as anemia. The red blood cell is quite a bit longer lived than the neutropenia. It tends to show up later if you're taking out just the red blood cell progenitor. But it can persist longer. If you are damaging megakaryopoiesis or platelet development, your toxicity is going to manifest as thrombocytopenia. If you're taking out T's, B's, or NKs, you're going to see lymphopenia. You may also see cytokine release as a result of therapy, and we've got good models to measure using whole blood to measure risk of cytokine release. And then a very hot topic of research nowadays, obviously, is ADCs. ADCs are linked to highly potent and toxic payloads. There's a lot of work around linker stability, trying to get the payload released in the target cell, but not released into the plasma, where it can cause all kinds of problems. And we've developed models, again, to look at the entire spectrum of neutrophil development. A lot of these ADCs they lead to neutropenia. So we developed models to look along the entire spectrum of neutrophil development, from the progenitor all the way through to the CD66B expressing neutrophil. And one question that often comes up is, are these drugs predictive for a specific modality, like small molecule versus ADC, or for a specific target, like CDK4/6 versus something like a PARP inhibitor? Most of these assays, because it's fairly easy to introduce molecules into in vitro models, most of modalities are compatible, at least theoretically, with these models. The question then becomes, are they translatable? So for example, we're going to show that there's translatable with tyrosine kinase inhibitors, but does that mean that there's translatability with ADCs, which is a very different modality and a very different source of toxicity? So it's important that we consider the modality itself rather than in addition to the assay when trying to decide what kind of assays to run. And on top of that, you need to think about where is the toxicity most likely to occur. So for example, if you have an ADC that targets a receptor on an early progenitor, like let's say CD33, which is on hematopoietic progenitors, then a progenitor assay, a colony assay might be suitable. If you've got something that's targeting a receptor that isn't on the progenitors, but may be showing up later in development, then the colony assay, the progenitor assay, might not be the best choice. You might want to look at more of a differentiation, like a liquid state differentiation assay. And the other thing is the stability and availability of the drugs in the assay system itself. If a drug has a high binding affinity for serum and the assay system itself has high serum levels, you need to wrap your head around that, see what the free fraction is, and we'll get into some models and point out the importance of understanding the free fraction that's actually floating around in the assay system in developing predictive models. All right, drilling down a little bit further into the hematotox assessment. There are three main lineages of cells that cause problems clinically when the drugs are affecting their progenitor cells. The neutrophils, which when you take them out, like I said, you can lead to clinical neutropenia. Erythroid cells, which can lead to clinical anemia, and megakaryocyte cells, which can lead to clinical thrombocytopenia when there's toxicity towards them. There are assays for all three of these. They're relatively straightforward. They've been around for probably 50 years. These types of assays were first developed for bone marrow transplantation assays. They were modified in the late 1990s and early 2000s and clinically validated to be predictive of toxicity. And they involve taking bone marrow cells, either purified CD34 cells or bone marrow mononuclear cells, plating them in appropriate cytokine formulations, letting them grow for about 2 weeks, and then counting the total number of colonies that grow out. And what you get as you dose in drugs in these in vitro assays, you get really nice dose curves that allow you to calculate IC50s. What we found over time, we started out offering the colony forming cell assays way back in 2007. They were already well established. The supply chain for bone marrow and for the reagents was there. The technical expertise existed. What we found over time was that the colony forming cell assays were really, really useful for a lot of programs to identify toxicity, but occasionally clients would come back to us and say, you know, we ran the CFC assays. The CFC assays didn't point to any, let's say, neutropenia risk or minimal neutropenia risk. But when we went to the clinic, we saw, you know, we did see neutropenia, or the specific case I'm thinking about was thrombocytopenia. So the question is, well, why did the colony assay, which is evaluating progenitors, why didn't it pick that up? And it turned out that the underlying toxicity mechanism was really aimed at the development of the mature megakaryocytes into platelets. And what the megakaryocyte colony assay does is it looks at the development of a progenitor from like a CD34 positive cell through to a phenotypically mature megakaryocyte cell in that it expresses CD41. It doesn't really tell you anything about the final maturation of the megakaryocyte into platelets. And this particular type of drug, and ended up being the class of drug, was inhibiting that. So we went and we developed some liquid assays with a flow cytometry readout that measure exactly that. We can demonstrate that our model, we drive the cells all the way through to CD41, CD42B positive Platelets, very small, forward scattered low on the flow cytometer, very small. And we can quantitatively enumerate the impact of drugs on the actual platelet development and the different portions of megakaryocyte development. All right. Combining that observation that different models are needed with what the FDA is saying about new approach methodologies, we really want to drive home that you need to cover the full biological spectrum of any given process, like blood development process, from a CD34 primitive progenitor all the way through to an activated cell. There's no one assay that's going to do all of that. You need overlapping assays. So here we've got 3 different types of assays. In the top row, we can start with, these are the colony assays. These measure the impact of drugs on the primitive cells. We take bone marrow cells or CD34 cells. We culture them with appropriate cytokines, and we develop and count, literally physically under a microscope, the number of colonies that grow out of each type, the erythroid, megakaryocyte, and neutrophil. We can rank the impact of drugs relative to each other. We can compare that to clinical benchmarks, and we can also marry them with in vivo mouse models to try to predict maximum tolerated dose in humans. And I'll get into a little bit of that later on in this. Like I said, sometimes the toxicity is actually after what we see in the colony. The specific example is with the megakaryocytes. The toxicity might affect the final stage development of megs to platelets, and the progenitor assay isn't going to see that. So we developed liquid phase differentiation assays, where we again start with CD34 cells. Typically, we drive them all the way through using cytokines to a mature phenotype. And we look to see if drugs are preventing full maturation, and we can rank them that way as well. Now, once the drugs are fully phenotypically mature, there's still a question about whether or not they're functional. Can they be activated? And so in those cases, we can take fresh whole blood. In this example, we're looking at platelets. We can isolate the platelets and then stimulate them in the presence or absence of drug and see if the drug is actually preventing upregulation of markers like CD63 on the platelets. So combined, we would consider those 3 assays to be the reasonable framework for a new approach methodology for evaluating hematotoxicity. Okay, so we've got the models in place, but the question is, well, do they correlate clinically? And our standard approach for asking that question is to run multiple drugs of a given class. In the current example, we're looking at tyrosine kinase inhibitors. We measure the IC50. So again, in this case, we're looking at the myeloid IC50s. TKIs had a lot of problems with neutropenia clinically. Well, some TKIs did. And we selected drugs that had clinical precedence. There was already clinical data on the drugs themselves. And we graphed the observed incidence of class 3 plus neutropenia against the IC50 in the in vitro assay. And what we found with TKIs, at least, was that the drugs that were least associated with severe neutropenia clinically had the highest IC50, meaning they were the least potent in the in vitro assay. And we went all the way through to drugs like dasatinib, which had quite severe association with hematotox and a very low IC50 in the in vitro assay. Information like this can help programs rank their drugs if they're looking at a lot of different drugs and trying to figure out where the toxicity is coming from. You can also use standards of care to anchor your tox if the goal of the program is to maybe hit the same target but develop a less toxic drug, which will in turn allow for a higher dosing of the patient. They might be doing that, so it allows for that as well. And then very soon we'll get into how you can actually use the data to try to evaluate or estimate maximum tolerated dose. All right. So this is what colony data actually looks like. This is for the megakaryocyte assay. When we count the colonies, we literally take the cultured cells, we dry them down, we stain them for CD41, which is a lineage marker of NK cells, and then we count them microscopically. We divide them into small colonies, those with 3 to 20 cells, intermediate colonies, 21 to 49, and large colonies, greater than 50 cells in the colony itself. And then we add all those up to get the total megakaryocyte colony forming cells. With, in this class of drug, these are PARPs. Olaparib, which is not really associated with clinical hematotox. There's some, but it's not considered a huge problem. It has a relatively high IC50 in this assay, indicating that, it's probably not much of a risk of hematotox. These aren't perfect, of course. They're not yes or no answers. It's more gauging risk. On the other hand, niraparib and talazoparib are both associated with hematotox. Talazoparib has a very low IC50 in these, and if you were to, look at the literature, you'd see that it is associated with a pretty high incidence of all three types of hematotox. What's interesting here is niraparib probably is not picking up as much toxicity. The assay probably is not picking up as much toxicity for niraparib, particularly in the megakaryocyte lineage cells, as one would expect. And what we think is that the colony assay, the toxicity with niraparib, it shows up more later than what you see with the colony assay. Again, the colony assay is looking at toxicity to primitive progenitors. It won't show up if a drug is, let's say, inhibiting final maturation of the megakaryocytes necessarily, or definitely if it's inhibiting actual platelet development. So I use this data set to show the good. You can rank the compounds, they do correlate, but also some of the weaknesses of just going with one model, or the definitive weakness of just going with one model. And you need to look at the full spectrum of development and make some rational, or ask some rational questions about where you expect the toxicity to occur. Okay. From there, you can actually take the IC50 values that we get in our in vitro assays and try to marry them with what we see in animal models for maximum tolerated dose, and attempt to predict the maximum tolerated dose in humans as a starting point for clinical trials. This is a very old paper, but I really like it because it was, it was the first paper that I was exposed to with this concept. That the industry has since developed a long way. So this is definitely not the be-all and end-all of marrying animal models with in vitro models, but it is kind of the first one that I saw. So I like to refer to it as, for training purposes. This is Noriko Masabuchi. Way back in 2006, he was trying to create, again, a model for estimating maximum tolerated dose in humans. He had three approaches. In his first approach, he looked at the maximum, he wanted to predict the human maximum tolerated dose by taking the MTD that was observed in mice and then adjusting it by the ratio of the IC90 in the CFU-GM assay. So he was particularly interested in myelotoxicity. The IC90 of the human CFU-GM assay with the IC90 of the murine CFU-GM assay. So the ratio of those two multiplied by the mouse would give, hopefully, a predicted MTD for humans. What he found was that the model was not overly predictive. It had an R-squared of 0.628. Some compounds seemed to cluster on the line, but other compounds were, clearly not responding to the model. Then he asked the question, well, maybe not the maximum tolerated dose. Maybe we need to factor in the time that the drug is actually in circulation. So he factored in the area under the curve at the maximum tolerated dose. Again, adjusted it by the ratio of the human IC50 value by the mouse IC50 value to predict the human maximum tolerated dose. That model also was not very predictive, even though you certainly suspect it would be. It seems like a pretty strong approach. But then he figured out that really you need to be looking at the free fraction of the drug and how long it's actually circulating at the maximum tolerated dose. So when he plugged those numbers into the model, incorporating the free fraction, what he got was a pretty good correlation between, there's that 0.887, between the predicted MTD and the observed, clinically observed MTD across a spectrum of camptothecin-derived compounds. Like I said, the industry has since moved on to much more sophisticated models, but I like this as a basis for starting a conversation like that. Okay, so from there, that's toxicity. There's all kinds of models. We need to be looking at the entire spectrum of development from the primitive progenitor cell all the way through to the activated cell. We can't be dogmatic and hang ourselves on a single assay, although certain assays like the colony-forming cell assay are very well regarded and well established for predicting toxicity. There are cases where you need to go a little bit further into the development with flow cytometry models centered around differentiation, starting from a CD34 cell, or even going all the way to like a whole blood model, isolating cells out of that and seeing what the drug is doing specifically to the fully mature cells, which you can only get, truly only get those fully mature cells from a peripheral blood draw. You can't make a fully mature cell in vitro. Okay, so from there, so that's the toxicity, but we can also do efficacy. And one great thing about the way the industry has developed in the last 20 or 30 years is that there are biospecimen companies like Discovery that will bank disease models, including blood diseases like AML, myeloproliferative neoplasia, sickle cell disease, all kinds of blood diseases. And we can take the models that we develop for the normal bone marrow to predict toxicity and modify them without too much work to ask questions around efficacy. So for example, we can take a colony assay and instead of plating normal cells and drug, we can plate AML cells and drug. The AML cells will form colonies that we can count and the drugs will deplete those colonies. So again, we can rank the efficacy of the drugs and we can further categorize the AMLs into different types. And a lot of the time the biospecimens come with matched plasma. So at the same time as you're evaluating your drug and looking at its impact on different patients, sponsors can run biomarker studies on the corresponding plasma to see if they can see correlations between markers that are in the plasma with efficacy in the primary cell, diseased cell models. The objective being that they'll be able to screen patients for, select patients more carefully based on biomarkers. And I mentioned plasma, obviously genetics is another important component of the patient screening and patient selection for drug studies. All right, there's all kinds of ways you can go about modeling drug efficacy using primary blood cells. Like I mentioned, you can take diseased cells directly. You can take AML cells for erythroid, polycythemia vera. You take cells from those patients, put them into models, and see if the drug can affect or reduce the EPO-independent development of erythroid cells. Similarly, you can run models for essential thrombocytopenia and myelofibrosis, looking at thrombopoietin-independent or thrombopoietin hypersensitive development of megakaryocytes, and you can look at the impact of drugs there. You can also develop models from blood, not necessarily from diseased blood, but you can take whole blood, or you can take, let's say, CD34s and develop them to mast cells if you're looking at immunogenicity or hypersensitivity. And then sort of create a primary cell model, which is as translatable as possible. from a Petri dish. Specifically, for example, for inflammation, you can take whole blood, add drug, and then stimulate it with something like Zymosan, and then monitor the impact of the drugs on the upregulation of neutrophils. And again, you can rank drugs that way, and then you can marry that data. You can take the same drug in a very similar assay and look at the impact of the drug on normal cell development and get that therapeutic window, the difference between the efficacy and the toxicity that will tell you if your drug is going to be more effective than it is toxic, which is a very important parameter when developing the drugs. And then the models also allow for a lot of add-ons. There's the primary readout. It can be by flow cytometry. We can harvest pellets to look at what's going on, let's say at an RNA-seq level. We can harvest supernatant to look at cytokine production. We can look at things like phosphoprotein. There's so many things we can do with these models to try to help the developers drill down on exactly what the drug is doing and which of their candidates is the most likely to succeed clinically. All right, and then just a specific example with megakaryocyte development. This is a model that we developed for toxicity. Like I mentioned earlier, the progenitor assay is great, but it wasn't picking up certain cases of clinical thrombocytopenia. So we developed a liquid phase assay where we start with CD34 cells, so blood progenitor cells. We put them into culture with thrombopoietin. They start to proliferate. We put stem cell factor in there as well. There's different models. Sometimes you add some different cytokines, but they're all variations on a theme. On day 3, they are proliferating, but they're not really differentiating. The x-axis here is CD41, which, like I mentioned before, is a marker on megakaryocytes. By day 7, we do see fairly significant development of megakaryocytes. And then day 10, again, even more. And by day 14, the assay sort of starts to break down. With something like essential thrombocytopenia, this entire process will happen in the absence of TPO. So we can modify the toxicity assay by dropping TPO from the media and then introduce drugs. And instead of asking, is the drug toxic, we can ask, is the drug effective at reducing this process in the absence of TPO in the appropriate disease models? Okay. So by way of a little bit of a summary here, we can take the human primary cell assays that we developed, modify them to look at the effect of drugs on hematological diseases and disorders. I've talked a lot about cancer, but sickle cell disease. We have sickle cell disease models as well. We can use the in vitro assays to ask further mechanistic questions, both around biomarker development. We can see in our liquid phase assays, at least we can do washout experiments to evaluate if the removal of a drug can rescue the progenitors. Some drugs are overtly toxic. They just kill everything. Some drugs kind of just inhibit cells at some stage of development. And when you remove the drugs, they'll continue on. And that can be very informative for when they're planning a clinical trial. Okay, and this is just a sort of an example of some of because of now the availability of diseased cells, some of the models that can be run. Multiple myeloma, marrow cells will form unique colonies when cultured under appropriate conditions, and we can evaluate drug impact on them with colony assays. AML cells, you can support them, you can support the progenitor AML cells and colony forming cell assays. There's also liquid culture models that have been developed, so you can look directly on the AML gate by flow cytometry and see if the drug is having an effect there. We can look at lymphoma models. More recently, we've developed models for polycythemia vera, essential thrombocythemia, and myelofibrosis. Again, genetically linked blood disorders. polycythemia vera, all of these had colony assays initially, and we are actively developing liquid phase assays to complement those colony assays in a similar manner to the liquid phase assays that we talked about for toxicity testing, where you can actually add the drug and then remove it and see what happens there, or you can feed the drugs into the assay system every second or third day if you're worried about stability. And then point here is that we're not just limited to anemia. We're not just limited to cancers. There's many other types of blood diseases that are specifically, especially the ones that are genetically linked, where we can develop a model of development in vitro and then start testing drugs in them. All right, and to summarize, the use of in vitro blood development models is inevitable. I think we're heading in that direction. We're working very hard to develop models every single day. The objective is to create clinically translatable models that allow decisions, including hard decisions, the program killing decisions, to be made earlier in the process. We're never going to get rid of mouse. We're never going to be able to recapitulate the full biological complexity of a living organism in vitro. But we do need to recognize that a lot of the newer modalities and the newer targets are only on humans. And so the animal models are going to be informative in some ways, but they may be completely uninformative, including NHP models in other ways if the pathways are really human specific. And so we need to continue to develop those predictive models that use human cells. Hematotoxicity in particular is a major risk. It's the first organ that just about every single drug sees, and it sees it at the highest concentration. So it's, you're often going to have hematotox associated with drugs. Primary cells have the potential, used correctly, validated appropriately, primary blood cell models have the potential to provide really strong, clinically relevant insights into the safety of drugs. And finally, mentioned it a few times, but I'll mention it one last time. The earlier we can ask those hard questions, even though it's a little counterintuitive sometimes and a little bit challenging to push, the earlier we can ask those hard questions, the better off the programs, oftentimes the programs end up being, because the resources, the heavy resources that are needed for the clinical trials are reserved for the really strong candidates, as opposed to pushing weaker candidates into a very expensive clinical trial, which then fails. All right. And with that, I will turn it back over to Bernadette for questions and answers.
Thank you so much, Gary, for a great presentation. As a reminder to our webinar participants, if you have a question for Gary, please type it into the Q&A box in the control panel. We'd like to ask attendees to take a moment after the webinar has ended to take our exit survey to give us your feedback. So let's jump into the Q&A. The first question we have is about antibody drug conjugates. And specifically, what types of drug efficacy testing have you done for antibody drug conjugates?
Sure. So those are typically, when we're working with them, those are typically targeting AML. We've done a lot of work on AML with those, both on the efficacy. So we can take those AML models, introduce the ADCs, and look at the impact on colony formation. There's also obviously a lot of associated concern with hematotox, particularly neutropenia, because the payload is going to get released, even if you release it into the target cell. Once that target cell lyses, there can be payload floating around in the space that then spills over, the bystander effect on other organs. So we do a lot of work on ADCs, both on the disease state AMLs and then the safety assessment.
Excellent, thank you. Question about what type of drug compounds can be evaluated for hematotoxicity? And I don't know, maybe it's better to phrase that the opposite way. Are there any types of drug compounds you would not recommend be evaluated for hematotoxicity?
Well, there's so many new modalities. Back 20, 25 years ago, there were small molecules and there were antibodies, and then ADCs were kind of emerging. But now there's so many different modalities. There's PROTACs, there's RNA drugs, there's drugs that are packaged in lipid nanoparticles. Most of those can be evaluated. We've got over 8,000 test compounds in our database that we've evaluated in different types of assays at this point. Most of them, if there's toxicity to be seen, we'll see it. Cell therapies are pretty challenging in these types of models. The cells themselves can be fragile, like a CAR T cell can be fragile. So it's hard to get it into, let's say, a semi-solid media, which requires vortexing, so you can inadvertently damage the cell itself. They don't have a lot of, or any motility really in a semi-solid media. So getting them actually to contact the target cell can be challenging, which leaves you with liquid format assays. By and large, the other modalities, let's say like a naked RNA, which I don't think anybody's even trying anymore, but something that might be sensitive to serum plasma, which might be broken down in the assay system itself by enzymes that might still be active in the serum. Those might not be predictive because you just don't have any active compound in there. What clients will often do is request a sample of the culture media. They'll request us to ship a sample of culture media down to them so they can evaluate the free fraction of drug in our culture media. So again, we can do those mouse correlation studies, the maximal tolerated dose type studies, and also look at the stability and make sure that the drug is actually still in its active form in our assay. So if we don't see toxicity, we're not just not seeing toxicity because the drug itself is degraded.
Thank you. A question on whether you're aware of any automated reading or scoring software for colony-forming unit megakaryocytes.
Yeah, years ago, I was involved in a project to automate colony counting for the three main lineages, erythroid, myeloid, and MKs. What we found, it's actually a commercialized system, it's been around for a long time. The system is really good at evaluating, or the systems out there, the algorithms, can be quite good at identifying healthy colonies. So you can take normal bone marrow, put it into the assay, grow out the colonies, put the dish in, and you get a perfectly valid reading that correlates with what you see under the microscope. The issue is when you start to introduce drugs, the size of the colony, the morphology of the colony, and even the density of the colony often starts to thin out. And that's where you're really interested in. That's like the sweet spot. That's the dose response curve. And once that colony morphology starts to change, the algorithms tend to break down. They start either misclassifying colonies or missing them. Or let's say you've got just some debris from some dead cells, they start getting allocated as CFUEs, which are small colonies in their own right. So what we've done, and we are currently, we're actively looking at systems, but we have never found anything that is better than sitting down under a microscope and counting the colonies, unfortunately.
Okay. Another question, what is your experience with PBMCs, peripheral blood mononuclear cells?
Oh, sure. Well, they definitely go with our focus on hematology here. A lot of our models are based around bone marrow mononuclear cells, specifically the CD34s. In a lot of diseases, the diseased CD34s are actually circulating in the PBMCs. So rather than, for example, with sickle cell disease, it probably, you'd almost certainly be unable to get IRB approval to draw bone marrow from a sickle cell disease patient on an ongoing basis. Maybe if they were undergoing a crisis, but you couldn't schedule them to come in to get their their bone marrow cells. But you could schedule them to come in through the appropriate clinic and get peripheral blood, 40 to 60 mils. We can then isolate their CD34s from the peripheral blood. Usually you'd isolate the CD34s from the bone marrow, but there are enough CD34s there. And then we can go forward with, let's say, an efficacy assay to see if a client's drug can stimulate fetal hemoglobin development in those CD34s as they develop into erythroid cells. So there's all kinds of models there. A lot of other, so you can do the same thing with myeloproliferative neoplasia. There's a higher pool of CD34s in the circulating blood, and it's a lot easier to get that circulating blood. PBMCs are often used with immunology type models where you are stimulating them and then seeing if a drug has the effect of tamping down that stimulation. You're either looking at efficacy, you want the response to be tamped down, or you're looking at some kind of toxicity where the drug is actually blocking the upregulation of something like a neutrophil. So yeah, there's all kinds of models that go along with the theme of hematology, experimental hematology, that we incorporate. Sometimes, we develop it intentionally. Sometimes a client will just come to us with an idea or a paper and ask us to reproduce it.
Thank you. Do you have any primary cell models for evaluating cytokine storm response?
Yeah, definitely. So there's a few ways to go about that. When you are looking at it, you can use whole blood models or you can use PBMCs. The full risk assessment, you look at, this came out of the follow-up to the TGN 1412 clinical trial, failure with severe toxicity from 6 donors that happened in the UK about 20 years ago. What they found was that they were missing, when they did their in vitro tox, they missed the cross-linking of the receptors. So they were targeting the receptor and seeing if there was an effect, but the toxic effect was actually due to cross-linking that doesn't happen. So as a result of that, there were 3 general approaches to assessing cytokine storm risk that were developed using PBMCs, which is to present the drug in a soluble format, just in with the cells, in a wet bound format where you coat a plate with the antibody, empty the plate, and then introduce the cells. That's wet bound. And then dry bound, which is where you coat the plate again, you empty the plate, but you let it dry for like overnight. And that the way that gets presented on the surface of the cell is slightly different. And in certain cases that's what you're going to see, specifically with like an anti-CD28 targeting antibody like TGN 1412. You'll see toxicity with that model, but not with the other 2 necessarily. Different modalities have different approaches. Again, you got to understand the literature and what is the current thinking of the key opinion leaders for assessing cytokine storm, but it's all based around some kind of whole blood or PBMC model.
Thanks so much. We're going to wrap things up for today. We'd like to thank Gary Dos Santos for his excellent presentation and our sponsor, Discovery Life Sciences. If we didn't have time to get to your question, we'll try to follow up afterwards. As a reminder, please look out for the survey as the session ends to provide your feedback. And if you missed any part of this webinar or would like to listen to it again, an archived version will be emailed to all attendees. Thank you so much for joining us for this genome webinar.